And then in 1999 came to UAB as chair of the Department of Biostatistics, a position he wisely held for only 12 years so that he could really do what his passion was. Actually, he was doing his passion while he was chair too, but so his career has really focused around biostatistical issues related to cardiovascular disease, particularly stroke, but he's really, I couldn't make it through all of the grants he's been a co-investigator on, but they've spanned a huge range. I think the thing that probably has defined his career the most has been the regards study. I remember exactly what
reasons for racial and geographic differences in stroke.
Okay.
You can't do science if you do academics.
Yep. Uh-oh. I'm finished. That's okay. And this has been funded since 2001, I believe,
yeah.
And it's funded through 2022, and will likely continue on if he can anoint the next person to be PI after that. It's been an incredible study, has national impact and involves multiple study sites. So one of the things he's particularly effective at is bringing huge multidisciplinary teams together and figuring out how to allow resources that were gathered for one particular kind of purpose to be extended to be used in other ways that really add value to the research that our community does. So I hope he'll take a minute to just talk about some of the issues around that kind of thing as he goes through his talk today. It may not be on his slides, but I think it's really important for you to realize that if you see an opportunity to use data in the regards study in something you're doing, you shouldn't feel intimidated to go talk about it, because there are ways to gain access to these kind of data as long as your rationale and stuff is all.
Well, I will take a minute and plug. This is not what I was going to talk about. And the nice thing about being a mathematician is I'm supposed to be absent-minded. It's expected of me, and I do not disappoint on this front. And so, but basically, this is a longitudinal cohort study of 30,000 people in 50 states, and we've been following them since DIRT. And the exciting thing for you is that we now have more than 100 funded ancillary studies to the parent study. And they're everything from, we have a biorepository, so it's genetic stuff and biomarkers to crazy things like Leslie McClure got a grant funded from NASA to do satellite measurement of air pollution at the people's homes. And so it's everything from blood in a freezer to satellites in space and any crazy ideas you have in between.
Great. Okay, so with that, I'll turn it over to Dr. Howard. And
But by the way, for the people at home, can you hear me speaking? Is this thing working? Or I'll keep talking unless somebody stops me. Anyway, I hope you can hear me anyway. I would rather have a chat than a lecture. And so I want to invite everybody at any time to stop me. This is going to seem a little bit disjointed, and the reason for that is what you're getting is, in Biostat, we teach really a 2-semester course in clinical trials that you're going to get in 2 hours. And so it's like a little bit of this and a little bit of that. And so the subtitle of this is sort of wandering through 1,000 remarkably different things in a 2-hour lecture. And so if you feel a little bit disoriented, or if I make a jump between two things too quickly, just stop me and we'll figure it out together. I want to make sure that you're comfortable with why this is important. I mean, basically, there's a spectrum of evidence in making medical decisions. And it starts with what are called ecologic studies. And there are 2 kinds of ecologic studies. As a clinician, you treat 20 people and you notice that something's going on and you say, gee, I wonder if this is associated with that. And the second kind of ecologic study are for example, people in southern France drink a lot of wine. People in southern France don't die of heart disease. Gee, I wonder if it's the red wine in southern France that stops people from having heart disease. Nothing was measured in an individual person. There were groups of people with one outcome and groups of people with a different outcome. And you just sort of wonder if they're associated. The second level, which I'd love to come back and talk about also, this is the regards part of it, is the observational epi study. And within observational epi, there's also a spectrum of evidence, the different level that you want to give credit to. The lowest level is what's called a case control study. I'm not going to talk about it today. The second level is called a cross-sectional design. And then there's the gold standard, the prospective cohort study of observational epi, which is considered the furthest up on the food chain for observational epi. And then there's the randomized trial, and everyone says this is the gold standard. And I want to take a minute and make sure, if there are 2 or 3 places in the lecture that I say, this is important. If you remember something, remember this. Okay, so this is important. If you remember something, remember this, which is why is the randomized trial the gold standard? And it's really interesting. Back in the 1990s, there were just boatloads of incredibly well done observational epi studies that said hormone replacement protected women from heart disease and stroke. To show you what an idiot you can be, I stood on the podium at the American Heart Association meeting with Trudy Bush arm in arm, and we said, you cannot do the women's health study. We already know the answer. This is unethical. We're exposing women to a treatment and we're going to kill them. And so I was anti-women's health because I knew that it protected women from heart disease and stroke. I was confident. Along come 3 clinical trials. One of them is the Women's Health Initiative. Much smaller than these huge and well-done epi studies. And they say that hormone replacement is not only not beneficial, but it might even be harmful. This, by the way, is the greatest teaching example of my career. Okay? You know, how can that happen? The entire field accepts the results of these 3 rinky-dinky trials, ignores tons of well-done epidemiology. You know, and how can this occur? Why do we accept the randomized trial? Now, this is the slide I want you to remember and the concept I want you to remember. In all of your work, you worry about 2 things in terms of making inference. One of those 2 things is called a confounder. And I don't know if you've gotten this yet in this course, but I want to tell you what I think it is that I have some risk factor, estrogen, and some outcome, heart disease risk. And what I'm interested in is the relationship between estrogen and heart disease risk. Now, that's nice, but I have some other thing that's messing up my ability to do this. And perhaps what happened with hormone replacement is this idea of socioeconomic status. Now, it turns out that rich white women take hormone replacement. You know, basically in America at least, who was on this drug were rich white women. Okay, and it also turns out that rich white women tend not to have heart disease. Okay, and so if you don't watch out, what you do is you think that the risk factor, oh, I can't point that way, that's right, I have people at home. What you find is that people, boy, this is very sensitive, is that people with the risk factor look like they have the outcome, but it might not be through this pathway. It could be that people on estrogen or high SES and high SES doesn't have the risk factor. So I need to take care of that confounder. Need to make it go away. I wish I had more time to talk about how you make it go away in observational epi studies, but you make it go away there. Okay? But basically the whole idea, by the way, I define a confounder as anything that's associated with the exposure and the outcome. Okay? My epidemiologist brothers and sisters sometimes say anything that's associated with the exposure and the outcome that is not in the causal pathway. Okay? I'm not smart enough to know what's in the causal pathway. Over and over again, I've been proven wrong when I thought I did, and so I've quit thinking like that. Okay? So basically, in this case, rich white women more likely to use HRT, rich white women less likely to have heart disease. So it looks like hormone replacement is less heart disease. So how does randomization fix the confounder? Well, with randomization, the high SES women and the low SES women are equally likely to be exposed. Okay, and why is that? Well, I've got a coin flip, and I'm going to flip a coin and give some women hormone replacement and other women not, 50-50 chance generally. And so basically that leg goes out. That leg is no longer there. And it doesn't matter that the confounder is still related to the outcome. Okay, doesn't matter. Okay, because for something to be a confounder, it has to be associated with both the exposure and the outcome. And what randomization does is make it so the exposure is not related to the confounder. Okay. Now, randomization is just the neatest thing ever for medical science. It is like a gift from God. It is wonderful. It is wonderful. Okay. Because not only are high and low SES women equally likely to get exposed, but there's equal exposure for all risk factors. And here's the key, even the ones you don't know about. So the cool thing about randomization is it fixes the problems you don't even know exist. And if you think, there are not many things in life that do that. That's sort of nice, okay? That's sort of nice. And by the way, it's so funny.
Requires the things you don't know about be prevalent at a level that they would fall within both of the randomization arms.
Well, they will by definition. In other words, let's say that I didn't know that inflammation was an issue. Okay, and so women with high C-reactive protein, women with low C-reactive protein, those people will be assigned equally to both groups also, even though I don't know it.
Things that could be quite rare might not. Seems like it.
Right. Well, if you're quite rare, the randomization might not equally allocate you.
Right.
But if you're quite rare, I don't care. Okay, because it has to be common enough to have an impact. And so let's say that it's women over 7 feet tall are at differential risk for heart disease. And we only have 2 of those women in my study, and both of them got randomized to receive hormone replacement. Well, I don't care. It's only 2 women out of a couple of thousand, Doesn't bother me. Okay, so why do we always do randomized clinical? By the way, that is the part I want you to remember. Let me back up right there. It fixes confounders, even the ones we don't know about. That's why it's the gold standard. So why don't we always do it? Well, to begin with, you don't start using with a sledgehammer. You start using a fly swatter. If you think that something might be related to something, you don't write a grant to NIH and say, give me $50 million. I want to do a randomized clinical trial. It's a process that goes through the ecologic, the epi. By the way, basic science to ecologic to epi to clinical trials. By the way, they're quite expensive. The one I'm doing right now is $67 million. It's written as an R01. And you don't get those things by accident. I mean, basically, and the key is all of that money goes to answer one question. Generally, in randomized clinical trials, you spend a boatload of money to answer one question. An advantage of observational epi is you get to answer multiple questions, not quite as well as a randomized trial, but you get to do a whole bunch of stuff. And some topics can't be ethically addressed by randomized trial. There's never going to, I believe seatbelts are protective. There is no 1A evidence that proves they are. We're not going to randomize people to get cars with them without seatbelts and see what happens to deaths on the road. It's so fascinating. I used to live in Winston-Salem. The tobacco companies say there is not definitive evidence that cigarettes are related to lung cancer. Turns out they're right. Okay? The definitive evidence would be a randomized clinical trial where we took children and randomized them to smoke and not smoke, and then followed them up to their. It is a brilliantly stupid thing for them to say, but you can't disagree with them. Okay. So in terms of the big picture, this is part of what David was talking about. Even within clinical trials, it's sort of a process. And so if you want to do a randomized trial, or if you're thinking randomized trial, they tend to come in 4 phases. So step 1, 2, 3, 4. Okay, just to confuse you, and if you're making notes, everybody defines these phases differently. And so you're going to go find, this, by the way, is the way people like Steve Hulley defined them. And so this isn't just me, but other people disagree with Dr. Hulley and me in terms of what is in the different phases. And so if you're reading something that's a little bit different than this, you can say, oh, that's what he was talking about. Different people define the phases different. So phase 1 is tolerance of the drug. First time in humans. Is this something works great in rats, works great in pigs? Am I going to kill people if I give it to them? And so usually phase 1 is toleration of the drug. Small group of people, in cancer particularly people who are with expected not very good outcomes, you give the drug just to make sure you're going to be okay. After that, you move on to early efficacy. Okay, phase 2, early efficacy, and that's broken into 2 steps. The first step is dose finding. Okay, and I wish I had more time to talk about this one too. Dose finding is really a cool topic. It's very hot right now in statistics. And the idea is that I know how much of this to give to a rat to be efficient. How do I take that and generalize it to a person? Do I just take the weight of a rat, multiply it times the ratio to weight of people, and that's how much I'm going to give them? So dose finding. The second step is early efficacy assessment, phase 2B. In early efficacy assessment, generally what you're doing is picking a surrogate outcome. So let's say that I want to see if something stops heart attack and stroke. Well, I don't have as an outcome of my phase 2B heart attack and stroke. I might have progression of carotid atherosclerosis as measured by B-mode ultrasound. I might have progression of coronary calcium as measured by MRI. Okay, and it turns out I don't really care how thick my carotid arteries are, as long as I don't have a heart attack. It's a surrogate outcome. They're using that because people who do have thick carotids do tend to end up with heart attacks and strokes. By the way, am I bothering you? Because I'm a wanderer. I feel like I'm behind you. You can have a crick in your neck before this is over. But anyway, so when you're a grant reviewer or when you're planning these things, the temptation of a phase 2 is to slip in and say, I'm going to use the outcome that I'm interested in. Well, if you're using the outcome that you're interested in, heart attack and stroke in the example I was just giving, and if you have enough people in your phase 2 study, that's called a phase 3 study, okay? In other words, if I'm adequately powered for the outcome that I'm really interested in, that's my pivotal clinical trial, okay? And most people, when they say a clinical trial, they're really talking about phase 3, okay? And in phase 3, we'll talk about this more. They're powered, they're designed for an efficacy outcome. I want to be able to see if I stop heart attacks and stroke. Good example, a great teaching example has been Vioxx, okay? Vioxx was powered to see if it prevents pain, because it is an efficient thing to stop pain. Now, it turns out in phase 3 studies, you have these other things called safety and regulatory requirements. And by the way, Vioxx, I've got NEOA a little bit, and I'll tell you, Vioxx works great. Really stops the pain. It does cause heart attacks, which is somewhat of a bad side effect, but your knees feel great while you're having your heart attack. Okay, and so that's the safety outcome. The challenge of a phase 3 study is that the spotlight is on the efficacy. Does it stop the process that it's being given for? It is extraordinarily rare for a phase 3 study to be adequately powered for the safety outcomes. Very rare. Okay. The corollary of that is drugs tend to get approved for use that are unsafe. Now we try really hard. We do monitor safety, and if there's a huge effect, you'll see it. But generally, it's not a huge effect. So then comes phase 4 studies, which are post-marketing assessments, normally with the focus on safety. Normally you do them for other reasons, but normally what happens is the FDA says, God, yeah, that looks really great. It really looks like it's working. It might have a little bit of a signal of heart attacks here, that we don't want you to have. Why don't you keep records for the next 10,000 people? You might have done a study of 1,000 people. Why don't you keep records for the next 10,000 people and monitor safety? Okay, so basically that's sort of the yes, ma'am. Does every drug undergo phase 4? No. Particularly things that aren't phase 4 is FDA land, and so a lot of things are not FDA trials. And so basically, if a drug is regulated, then the formality of safety becomes more of an emphasis. And so things that are FDA are more likely to end up as phase 4. Yeah.
As physicians, when we report
I'm hard of hearing. What?
As physicians, when we report adverse events,
Uh-huh.
as physicians, when we report adverse events, does the information get then transmitted to?
Right. Well, the question was, if you're holding the microphone, I don't have to repeat it, right? Okay. So so basically, yes, they go into a system. I'm having a senior moment right now that's run by the FDA. Does anybody remember what it is? I know it. But anyway, basically, it's a bunch of garbage. And the reason it's a bunch of garbage, in my opinion, is that these things are not, yes, you're supposed to do this. Do you always do it? And you always do it in the same way as a clinician? And the answer to that is, even if you did, your brothers and sisters don't. And not only that, normally it's unblinded. Again, Vioxx is a great example. If you're doing a study on Vioxx, a safety study on Vioxx, and somebody says, this could cause heart attacks, then you're very likely to report heart attacks. In fact, you might even over-report heart attacks. But if it causes hair loss and nobody tells you, you think, oh, the guy's going bald, There were other questions? Yes, ma'am.
Yes, Dr. Howard, I had a question.
George, by the way, I'm old enough without your help.
George, about phase 3 and phase 4, you were saying how phase 4 is that normally FDA land.
Uh-huh.
Was that the case with the hormones for women, postmenopausal women?
No,
was that phase 4?
That was a phase 3. The Women's Health Initiative, if you don't know about it, is a great read. Go get the paper and read it. It is fantastic. And the players in it, it was so much like, it was like, reality TV. It was just an amazing process. And a heroine in this is this woman named Elizabeth Barrett Connor, who is just the neatest woman in the world. Okay, neatest person in the world, I should say. And she's one of the people who really pushed for it. And so what she said is, it's nice that you think this, but this is not definitive evidence, and it could be confounding. And so actually, Women's Health Initiative was a phase 3. It was done as a phase 3, and there the efficacy outcome was, does this stop, among other things, heart attack and stroke? It also did bone loss and other things. I think there were 4 outcomes in the study, if anybody remembers.
Does the FDA actually call for phase 4 studies for specific things, or is it investigator-initiated generally?
It is generally FDA mandated. We will approve you if and only if you're a good boy and do a phase 4 study. And so, because they're expensive. And and these companies, for better or worse, are in it for the money. And so they don't do these things by accident. Okay. So moving on, this is the outline of what I'd like to talk about. If I don't make it to the bottom, I'm sorry. David will invite me back. We'll finish it later. But basically, each one of these, by the way, is a 1-day lecture in the course. And so we're going to go through this like a thing. And so basically, you have to take the hypothesis carefully. And the key to a clinical trial is this concept of equipoise. And a lot of people haven't stopped to think about what equipoise really is. Now, it's a general uncertainty in the medical community over which treatment will be beneficial. By the way, I didn't say that you're uncertain. I said that the community's uncertain. How many surgeons are in here? I love surgeons. I work with surgeons all the time. Surgeons never doubt. And if you stop and think about it, if you're cutting on somebody, I think you really got to believe you know what you're doing and you're saving their life. I don't think that's bad. I think it's a personality. I've never met a surgeon that's equipoise. I've never met a surgeon in these surgeries. I do randomized clinical trials of endarterectomy and stenting, and they know that they are saving people's lives when they do an endarterectomy. Turns out they were right too. Okay. The reason they want to do the trial is, damn it, they're going to show everybody else this thing saves lives. And I think that's cool. We need to celebrate that. I mean, that's okay. That's okay. They don't doubt. Okay, they are not at equipoise. The medical community is at equipoise. Now, that sometimes could put you as a clinician in an uneasy situation where you say, I don't really want to randomize this person. I know I can help this person, which is a very awkward situation to be in. I can't even imagine. And that brings up, and so basically, equipoise versus the uncertainty principle. Any Europeans here? No Europeans. Good. In Europe, they don't think equipoise, they think uncertainty principle. A slightly different concept. They say, as a clinician, we want you to put people in the study that you don't know how to treat. Okay, that you're uncertain how to treat. Now, the people you put in a study might be different than the people you put in a study, because the things that make you uncertain might be different. And so that's one of there are several differences between European trials and US trials. But generally, the goal of the US trial is every eligible person is going to be put in my study. And you monitor this and you say, oh my God, you're not putting eligible people in the study. You could be biasing the study. Okay? I think if we're honest with ourselves, even in America, we sort of slip into doing uncertainty principle. I hope that if I'm your patient and you're really uneasy putting me in a trial, I hope you don't do it, even though I might be eligible. You know, I came to you because I really believe that you know what you're doing and you've read the literature and you're a smart person. And if you're really uneasy, please don't put me in your trial. And so I don't think it's, to me, people poo-poo the uncertainty principle. I really think we do it too, we just lie, okay? We might do less of it. Second idea is informed consent. And basically informed consent is really a hard topic, by the way. Again, I wish I had more time to talk about informed consent. The idea, how many of y'all have all consented? Many of you have consented people. Deep in your heart, tell the truth. Do you really think they understand what you're talking about? And, I mean, this is, hit my hand. Welcome to the lecture. This is to protect the university, not to protect the patient. And I didn't say that, okay? But basically, it's so funny, regards the study David was telling you about that I'm part of, the most invasive thing we do in that is draw blood. That's our informed consent is 8 pages long. And all we're doing is, the most invasive thing we're doing is drawing blood. Okay, so the hypothesis, you need to be careful with this. When you're doing it, if you're writing an hypothesis for a study, go get a textbook. Every textbook will have a list like this in it, and make sure it's just like a checklist. Don't do it without a checklist in front of you. Okay, it needs to be clear, quantifiable, simple, focused, singular, and it needs to say the who, what, and when of the study. Okay, don't do it without a checklist in front of you. You'll mess up. Okay, this is the hypothesis for CREST, which is to contrast the relative efficacy of carotid stenting versus endarterectomy in preventing stroke, MI, and death during a 30-day peri-procedural period, and stroke ipsilateral to the study artery in the extended 4-year follow-up in patients with symptomatic or asymptomatic extracranial stenosis. If you go down that checklist, that hits all of those things. And it says the when, it goes over it in detail. Okay. Now, this is another hot topic frequently misunderstood. Yes? Focused is, they're number one, they're first cousins, okay? And so, but the idea of, to me, singular, I'm going to lie to you. This is a lie. Singular means you want to keep your eye on one and only one ball. In other words, I'm going to have one outcome. It's going to be assessed at this point. It's going to be done exactly like this. That's also focused too. Okay, but the idea of single, and now the reason I'm lying is there are trials called 2x2 factorials, and if we get there, we'll get there. But you can do randomized trials. I need to come back so you can. But anyway, and I forgot to repeat the question. The question is, what was the difference between singular and focused? I'm supposed to repeat your questions unless you're holding the microphone. And focus just means, who's eligible. I mean, you don't say I'm trying to prevent stroke. But I mean, it really has to be specific, and it'll come back and help you. So going back to superiority versus equivalence, this is again a topic that, by the way, I'm changing topics here, jumping between topics. So there are hypotheses that are superiority hypotheses, and there are hypotheses that are equivalency hypotheses. And to begin with, most trials, and the ones that make me happiest to be part of, are superiority trials. And so what you're doing is you have two treatments generally, and you assume that they're the same, and then you try to prove that you're wrong. By the way, the way statistics works in general for everything is you make an assumption that you think is incorrect, and then you try to prove that you were right, that it was incorrect. So if I think this new drug helps somebody, I start off by saying, no, it doesn't. Okay? And then I try to prove that the data don't support the no, it doesn't point. That's a superiority trial. So there's an old treatment and a new treatment. And what I'm trying to do is show that the new treatment, slight change, but suppose there's a new treatment with fewer side effects and lower cost than the old treatment. Well, I could just want to show that the new treatment is as good, that is really not worse than the old treatment. A good example of that is endarterectomy versus medical management of carotid disease. I mean, all things being equal, I would like to avoid the surgery. If the medical management of extracranial disease is as good as doing surgery, I'm not going to be doing surgery. Okay, that is not a superiority hypothesis. There we're, oops, I'm going the wrong way. There we're slipping into the idea of saying, I want to prove it's as good. Now, there are two reasons why any experiment can be negative. Okay, the first reason is the drug doesn't work. Okay, and the second reason is I didn't have enough people in my study to see what the drug was working, was really doing. Okay. Unfortunately, if you have a study that is non-significant, you can never know for sure which one of those two things are happening. You can't look at it and say, I bet the drug really works. I bet I didn't have enough people. You shouldn't do that anyway, but you shouldn't do that, okay? The corollary of that is if the study is positive, I can make a definitive statement. The drug works, but if the study is negative, I can only fail to reject my null hypothesis. I either accept my alternative. This, by the way, is one of the important things in all of statistics to get in your head, where I fail to reject my null. And that implies that traditional studies cannot do this. They cannot do this, proving that it's as good. A traditional study cannot do that. And by the way, one of the things that you should train yourself to never say is if a trial comes out negative, never again, as long as you live, say the two treatments were the same. And people say that all the time. You did not prove the two treatments were the same. You just failed to prove that they were different. And that sounds like it is a double negative. It sounds like a, kiss your sister kind of statement to make. Okay. But you cannot say that they're the same. You've not proven that they're the same. Okay. Now, so let's say that what I do want to do is prove that they're the same. I want to prove that this new treatment is as good or better than the old. Well, how do I do that? And the answer to that is I change the form of my hypothesis. I ask a different question. Specifically, this was invented by Blackwelder back in 1982. And basically what you do is instead of the null hypothesis being the two treatments are the same, I make my hypothesis. By the way, the first time you hear this sounds so backwards. Okay, you make your null hypothesis that the new treatment is worse than the old. I assume that they're different And specifically, I assume that my new treatment is worse than my old. Okay. Then what I do is I make my alternative hypothesis that it is as good or better. Now, what is the strength of this? By the way, I play the same statistical game. I either accept the alternative hypothesis or I fail to reject the null hypothesis. But now I've set myself up so that if I reject my null hypothesis that the new treatment is worse, I can now accept the statement that the new treatment is as good or better. Same statistical game, I've just asked a different question. Okay. Now, so now if we reject the null hypothesis, I get to make the cool statement that is as good or better. And it's simple in concept, and there are lots of names for this: non-inferiority, equivalency. Some people just say a Blackwelder trial. Okay. So regardless of the form of the hypothesis, the study still provides an estimated difference between the two treatments. If you really come down to it, and as you become more comfortable with statistics and more comfortable with trials, start looking at the estimate and the 95% confidence bounds around the estimate. Okay, start looking at the estimate. You know, it's so funny, if David does a study and he ends up with a p-value of 0.04, and I say, God, that's exciting. I'm going to replicate David's study. And I come up with a study that with a p-value of 0.06. Have I refuted him or have I confirmed him? You know, how would you interpret that? He said that there's a 4% chance that differences like this would have happened by chance alone under the assumption of all the assumptions I had in terms of the distribution of the outcome. I come out and I say it's 6%. I think I've confirmed him. All of that is because I can focus on the confidence bounds around the estimate. And really, that's I think we're moving away from p-values. We're moving away from point. Do y'all know where 0.05 came from? This is a great story. You know, somebody asked R.A. Fisher, who's like God of statistics, how much evidence does it take for you to reject a null hypothesis? And he says, gee, I don't know, about 1 in 20 does it for me. And out of that comes, 0.05. Okay. So basically, the link between the hypothesis and estimation, if the hypothesized difference is within the 95% confidence limits, I will always fail to reject. In other words, if I assume that if I want to test if this is different from 0, if my 95% confidence limits include 0, I will not reject. Okay. On the other hand, if the hypothesized difference is outside of the confidence limit, I will always reject. So if the lower limits of my 95% confidence limits don't include 0, I will always reject. Confidence bounds and hypothesis testing are tied, one and one. So basically, so let's say I have two treatments or products, and I have an established treatment and a new treatment, and I have a dichotomous outcome, percent of people who die, something. And so I have the percent that die with the old treatment and the percent of people who die with the new treatment. And in general, which treatment is best. And so basically, if I do a traditional trial, I'll say I'm going to assume that there's no difference between the treatment groups. Do I need to do something?
I'm just fixing some audio for you.
Okay, thank you.
Sorry.
Okay. So pay no attention to the man behind the curtain. But anyway, so basically, I assume there's no difference between the two groups, and so my null hypothesis, this no longer works. But there you go. Oops, now it works. So basically, my null hypothesis is those two proportions are the same. I say, what is my alternative? Is that they're different. Okay, and what I want to do is test differences between the group in a superiority, the traditional approach. And basically, so what I would do, here's my old treatment better, new treatment better, 0 is no difference. So basically, step 1 is I calculate the difference in the proportions, and I've shown that with this, hypothetical example of that orange bar. And I calculate 95% confidence limits around that interval. So now those are my 95% confidence intervals. Okay. And basically, if the 95% confidence limits do not include 0, then the two groups are different. So if you do this, there are three things that can happen. I can have the new treatment better. Okay, I can have no difference between the treatments or no evidence of a difference. See, I slipped and I said it too. Okay. Or the old treatment is better. So there are three outcomes that can happen in a traditional trial. And that you should know that. You should be cool with this. So what happens if we, so what are the advantages and disadvantages? The advantage of a traditional trial is I can definitively state if one treatment is better than the other. The disadvantage is if there's not evidence of a difference, I can't say the two treatments are the same. Null hypothesis never accepted. And you end up saying there's not sufficient evidence in these data to establish that a difference exists. That's what you're supposed to say, not that they're the same. Okay, but sometimes, as I've said, the manufacturer or the drug company wants to say that something is at least as good. And then you do an equivalency analysis. And here again, goal is to say, really the goal is to say it's as good. The better part would be nice. So basically, the null hypothesis is the new treatment is worse than the old plus a fudge factor. Okay? So basically, you say my null hypothesis is the rate in the old plus a fudge factor is going to be less than the rate in the new. I'm sitting here pointing at that like it's projected. Sometimes I'm not real smart. But anyway, and then the alternative hypothesis is that the new treatment is as good or better than the old plus the cushion. Okay, so you might be saying right now, where do you get the cushion from? Where does that delta come? How do I pick delta? How big of a cushion do I need to have? And the answer to that is that, first of all, as good generally means somewhere within the range of this cushion. Usually, like in my world, in cardiovascular disease, FDA usually says within 2%. It's the same. By the way, the drawback of which I'll talk about in a second, of a inferiority trial is at the end of the day, you have to defend your delta. You have to say, I think as good it means within 2%. David might come back and say, well, no, I think 1% is as good. So then you have to argue this point, which is, so basically what you do here is that this is the way this is set up. So you have, here's the zero and here's the delta. And I do the same thing. Step one is I calculate the difference in the proportions. And there it is. Okay. I calculate, this is important, the one-sided confidence bounds around that difference. I'll talk about one-sided versus two-sided soon. Okay. So basically what I do is I say it's somewhere in that range. And basically if the 95% confidence limits do not include delta, zero is not what you're after. It's whether or not this gap is there, right there. And so if the 95% confidence limits do not include delta, then the new treatment is as good or better than the old. Now I want to take a minute and make sure you're comfortable with this, because non-inferiority trials are getting really common. And you might say, why are you doing that? Well, basically, if I do a study and I get exactly the same, what I hypothesized, I have two treatments, I do the test, and to the 83rd decimal place, they're the same. So that would say that this point right here, this orange point, is at zero. That orange point, that would be what would happen if I got exactly the same answer. Well, if I get exactly the same answer, don't you think I ought to be saying that they're the same? Okay? But there's uncertainty about that estimate of zero. In other words, if I repeated the experiment, I'd get a little bit different, Okay? And so basically this fudge factor here is saying that this accounts for the uncertainty. This delta accounts for the uncertainty about the estimate, so that if this is right at zero, this will be right at delta. Okay? And that's the way you ought to be thinking about how to pick delta. Yes, sir?
Now, do you need to predefine the delta?
Absolutely. The question, by the way, I need to get in the habit. The question was, do you predefine the delta? Yes, you do. It gets written in the statistical analysis plan. It's literally chiseled in stone.
How do you come up with a little delta.
Okay, the question was, how do you come up? Oh, that's right. You got to train me here. But anyway, the question is, how do you come up with a reasonable delta? To begin with, if you're doing a sample size calculation, which we'll talk about in a little while for a regular trial, you have to define something called a clinically significant difference. What size difference would you take to think that your study would change the practice of medicine? Okay, clinically significant difference. By the way, we call that capital delta instead of little delta, but it's really the same thought. This delta is, how big would this have to be? You know, like if it was not really zero, but it was minus 3%, God, I just can't call 3% the same. I can call 2% the same, but I can't call 3% the same. Now, you should feel uneasy about this, okay? You should think, oh, God, that seems stupid. And if you think it's stupid, go figure out a better way, because this is the best we got right now. So basically, it's very tightly tied to the concept of what is a clinically significant difference. And what we're trying to do is pick a delta that's sufficiently small that says, even if I was off by that much, I don't care. I don't care. The question of how you get it, if it's an FDA trial, it is a matter, you have a pre-protocol meeting with the FDA and it's negotiated. And basically, you know, the FDA says we'll accept it as the same if it's less than 1.5%. You say, yes, sir. Okay, going on. So what are the outcomes of an equivalency testing? It is either as good or better, because this, whoops, back up. It's either as good or better because this thing did not include delta, or it is not as good or better. So you only have two outcomes in an equivalency trial. So what are the advantages of an equivalency approach? The big good news is you get to make the definitive statement is as good or better. The statement largely meets the needs of the manufacturer. And the disadvantage of it, which I said, and I wish I'd have had on this slide, is you're stuck forever defending your delta. Okay? In fact, if you're doing an inferiority trial, I would encourage us all to get in the habit of saying it is as good or better. And by the way, I define that as within 2%. So you make that part of the same statement. That's not true in the superiority trial. In the superiority trial, I have the capital delta that I use to make the power calculations for my study. But if I say they're different, it's not dependent on what I thought the difference was. It's dependent only on the data, so I don't have to defend that delta.
George, you keep mentioning I can't say it. Inferiority.
Non-inferiority.
Can't say the basis trial. So I was wondering about social behavioral science.
Mm-hmm.
Those trials. Would you take the same approach?
You could, absolutely. Let's say that what you want to do is you're talking about treatment for alcoholism. Okay, and you have something that, and the standard is something that would be burdensome. Patient has to go to 5 meetings a week. They have to take pills that they throw up if they, I'm making this up, I don't know, but help me here. Take pills that if you drink, you throw up and feel bad. And I've got this new treatment, yoga. And it's sort of fun, makes people happy, and it might stop you from having from drinking, might prevent alcoholism. Something that's nice and warm and fuzzy, and I'd like to do it anyway. Okay? Well, if that is as good or better than this other thing, I don't need to prove it's better. If it's as good, I'd rather do the yoga. So basically, the whole idea of a non-inferiority trial is if the new treatment costs less invasive, less intrusive, or if it makes a company a ton of money, then you might be doing non-inferiority. I will tell you, in all of my life, I've done one non-inferiority trial, and I was unhappy for the 6 years it took me to do it. Okay. I don't like it. You see it. It's going to come. It's like, you're going to get them. It's coming. But that doesn't mean you have to like it. I don't like it. I like superiority. I like saying these things are different. Yes, ma'am.
In your experience, is it easier to prove non-inferiority than superiority though? Because I feel like the majority of the clinical trials, at least that I've read in the last 2 years, have been non-inferiority trials.
Really? I haven't. The question was, what was the majority? Which one of these is the bigger pot? Is sort of what the question is. Are people doing more non-inferiority or more superiority? It'd be an interesting thing to do. It'd be a fun paper to write. I think Journal and a few others, and just to count the numbers both ways. I have not done that, but I would guess that 90% are superiority. But it could also differ by medical field.
Yeah, I'm in oncology, so I feel like in the world of oncology, kind of speaking to what you were just saying, a lot of medicines have some effect, and so a lot of times we try to prove non-inferiority compared to playing standard, and maybe the side effects are less, so we would choose to approach versus the standard care approach because less side effects with non-inferior outcomes compared to standard.
And a corollary of your point is the meaner you, and I don't mean that for you, I mean it of what you're doing to the person. The more distasteful the treatments are for somebody, probably the more likely you could be going non-inferiority. And if I could avoid chemotherapy, this would be good. Okay. So one-sided versus two-sided hypotheses. And so basically I have an old treatment and a new treatment, with events. And, I can either assume that there's no difference and try to prove that there's a difference, or I could say that there's no difference and I could try to prove that one treatment is better. This is both in superiority testing. In other words, let's say that I want to do a superiority trial of in my world, endarterectomy versus medical management. Do I really need to prove that medical management is better than surgery? In other words, I care if surgery is better, but if I fail to reject, and say I can't say surgery is better, there's no evidence that these two treatments differ. Is that sort of good enough? Do I need to say no? Medicine's clearly better than surgery. And so that's the one-sided hypothesis. So again, assume the new treatment would not be used unless it's clearly better than the old, and that would be the surgery is clearly better. I'm pushing this. I'm a little bit behind, so I'm going to skip over this. I think you know what I'm saying. So why would you use a one-sided hypothesis? Well, the reason you use one is that you're restricting the alternative hypothesis. You're sort of not looking on that side and putting all of your power on the other side. Okay, I believe I'll use the arrow key. That's going to hold me back. Okay, so and so basically the idea is that if you do use a one-sided hypothesis, you can shrink the size of your study. In the case of this example here, 25% event rate, 80-20 difference, you can see you save about 20% in your sample size, and this is good. By the way, it's also an ethical issue. That's also 150 people who I haven't needlessly exposed to randomization. And so if you're not interested in the increases in surgery, then this 21% reduction is great. So when should you do it? And I'll argue you should never do this. I mean, don't do a randomized clinical trial with a one-sided hypothesis ever, 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 as long as you live. And the reason for that is because you're providing valuable proof. If the one-sided hypothesis is used, you cannot definitively prove that surgery was inferior. And I keep picking on surgeons. I'm sorry for the surgeons in the room, but unless you can tell a surgeon that this is harmful, they're going to keep doing the surgery. I mean, it's just the way it is. So and also the one-sided hypothesis smells bad. When you write it up, they'll go, was that a priori one-sided, or did you have something that was almost significant and then you decided to do this afterwards, and it just gets you in trouble? All of that was the hypothesis. Boy, we've got a long way to go. Why don't we all stand up and stretch? And I'm not going to make it anywhere near the end of my talk. Just about a 5-minute break, not much. I feel time urgency here. I appreciate the questions, by the way. I'd rather not finish and have questions than finish and not have questions. What?
This is great.
Oh, thank you. I was worried it was a little bit too abstract or something.
Well, I don't think people can follow your formulas,
Uh-huh.
which come up unless they've had statistics than most people have. But the concepts are straightforward.
Yeah.
Good. It's great.
Well, clinical trials are simple things.
Well,
It's just hard, simple things that are hard to do. Easy to, in concept, very easy to do. As long as everybody does what they're supposed to do, they're even easy to do. Hey, George, good to meet you.
I'm in neurology department. Doing mainly neuromuscular therapy.
Oh, cool.
Yeah, but I came down from Cincinnati.
Oh,
My stroke was really big. So.
yeah, no, but Casella is a dear friend, and Kleindorfer, and
Yeah.
Matt Flaherty, and basically that's my second home.
Yeah. So Joe Broderick really found the department with the stroke.
Uh-huh.
He's still very strong in research.
Right.
So stroke's always on the side of my brain.
Yeah.
But I'm into neuromuscular.
Oh, cool.
And I was wondering, the regards data, do you also collect other diagnoses of the participants?
We have a self-reported hospitalization.
Okay.
Now, I don't know if you've seen what people say they get put in the hospital for, but it's really crazy. So the answer is probably not well enough to really do it.
So you don't know if they have Parkinson's, if they have...
Now we actually have one Parkinson. What is the woman's name in neurology here?
Utopia.
Yeah, yes. She has written one Parkinson's paper out of regards.
Does that mean that the patient's other diagnosis has been followed as well?
No, that means that she had a list of medicines that you only take if you have Parkinson's disease.
So their medications.
So we know their medications. And so what she did was, and you know what they are, and I don't. But I sent you the paper, or you can find it. It'd be easy to find. But yeah. And so what she did.
The reason I'm interested in, I take care of myasthenia patients.
Uh-huh.
Myasthenia gravis. There's one particular medicine that everyone takes and no one else takes.
Right.
It's a medicine that's pyridostigmine.
Yeah.
So I wonder if I can identify similar kind of cohort within the regards data.
I'm sure you could. Now, we only have medicines twice. We have it at baseline and then 10 years later. It sounds like you would think getting medicines is easy. It's really hard.
It changes all the time.
It not only changes all the time, but I know I'm on a statin. I'm not sure which one. And I mean, I should, and I'm pretty informed. And if you ask, the person on the street, what do you take? It's a lot of. And so what we've done is go into their house, gotten the medicine bottles out, put them on the table, and written down what they were taking. But you'll be amazed how many ways you can spell aspirin. Aspirin, I mean, there were, I think, 380,000 unique spellings of drugs that got sorted. I mean, but I'd love to sit and talk to you about it. No, it'd be great.
So yeah, my question is, because they have, because they part-time moving, they're very overrepresented in the obesity and also metabolic problem like hypertension, diabetes.
Yeah.
So I'm wondering if they have higher risk of having cardiovascular.
We could do that. The question is what proportion of the population, because we are pretty population representative.
Yeah, it's about
So
1 in 5,000.
Okay, so that means that with 30,000 people, we would have 5 or 6 patients.
Well, it's a pretty small number.
Yeah, you just got screwed. Hate to push, but let's get started. Is anybody out there? What? Okay, let's get started again. So where we're at.
I'm sorry, I'm going to interrupt for 1 second. So you should have gotten a notice that Friday from 9:30 to 1 o'clock, there will be a bioethics forum that the CCTS organizes. It should have come to everybody. Some of you we invited, so we know you work in the area where this is important. So the topic is on the role of trust in successful recruitment of patients in large genomic, for example, will be used as one of the examples. So if you're interested in that, you'll be sure to have a lunch if you register. There will be links. There are links on the CCTS homepage for this, and I encourage anybody who has an interest to come. It really will run from 10 to 1, but the noon to 1 is a lunch hour that's really sort of planning for future sessions. So the sort of the meat of the discussion will be between 10 and noon. It's going to be held in the Medical Towers, which is the building where the Division of Preventive Medicine is located up on 11th Avenue. It's up the hill, and it's about 17th or 18th Street.
It's at the top of the hill.
It's at the top of the hill. If you need more information, let me know, but please register if you're interested.
Thank you. Okay, so we are at eligibility now. We're making some progress, not as much as I hoped to. And so basically the next question is, who do you want in your randomized clinical trial? And it turns out there's sort of competing demands here. The first idea is you want to get a homogeneous population. You want to get a homogeneous population. And so basically any divergent subgroup of people, weird patients in your study can screw you up. And so you want to keep weird people out of your study. The trials that we do in stroke, we set a lower age limit for stroke at age 45 generally, because people who have stroke below age 45 is sometimes not an atherogenic process. It could be related to drug use or other things. And so basically we don't want to have people who are having stroke for strange reasons in our study. Okay. The restriction of the population removes noise from your study. You can always overcome noise by having a bigger sample size. But the more focused your study is, you remove the noise, your sample size comes down, you're more likely to get funded. Okay? The restriction of the population to the homogeneous group of people that you want to use is a great thing. Alternatively, you want to have your study generalizable. Okay? And basically, at the end of the study, you are not going to be able to really, if you're telling the truth, say, this is what I found and it applies to David, unless David was eligible for the trial. So I want to have this as broad as possible. I want to be able to get to the end and make statements. An interesting thing in trials for years and years and years, we excluded women from trials. Mr. Fit, why did we exclude women from trials? Seems like a crazy thing to do, was a crazy thing to do. But the reason for it is I want to increase my number of events because that lets my study be smaller. And if I only study men, I'm going to have more heart attacks and strokes than if I study women. And so I can have a smaller study if I study men. It's good. Small studies are good. And I bet women are the same. They might be. They might be. But anyway, but at the end of the study, you want to be able to generalize your findings to everybody who has the disease. And the question of generalizability, everybody you excluded from your trial, you really shouldn't generalize your findings to them. And having a broad inclusion criteria, welcoming all, is a good thing. well, clearly those two things are pointed that way. I mean, there's no way to have a homogeneous generalizable population. And so what's the correct answer? And obviously the answer is there is no correct answer. And so what you have to do is a balancing act. You have to think how weird are the weird patients. You know, you don't want the really nutso people in the study. And the example of that again is if you have a stroke and you're 30 years old, if you're white, chances are it's not because you develop atherosclerosis and lipid lowering is not likely to be very efficient in a 30-year-old person. The results accepted in all subgroups is there really one disease. You know, you want to, if there are multiple diseases, stroke is a good, thrombogenic versus embolic. You know, you have to think what you want to do is there are really multiple diseases. And which subgroup analysis will be required under any condition. And basically this is, you have to use good judgment. You have to stop and think, but do stop and consciously think. I want to be homogeneous. I want to be generalizable. Where do I draw the line? Where do I draw the line? And it's difficult to depend on subgroup analysis. I'm going to talk about that in a second. On the knee jerk is to say I'm going to include everybody, but then I'm going to analyze, I'm going to analyze people with AFib as a subgroup because they might be embolic strokes. And I'm going to analyze people with extracranial atherosclerosis because they might be thrombotic strokes. And that way I can understand what's going on in embolic and thrombotic groups. You get in trouble with that, and I'll show you. Which really introduces us to subgroup analysis, which is the next thing on the agenda. And this, by the way, is absolutely natural. It's absolutely natural at the end of a trial to say, are there subgroups of people who this works better on than other people? Natural question. Are there subgroups where it's particularly efficacious or particularly non-efficacious? And these are natural questions. We need to plan for these. We need to think about these before we get started. And at this point in the course, there's always one of the students when this is done in slow motion who raises their hand at this point and says, but you said confounding is not an issue. You said these third factors are not important in a randomized clinical trial. And this is not confounding. This is effect modification. In other words, I'm not saying this third factor is in the pathway of action. What I'm saying is this third factor separates people into two groups, and those two groups have different responses to the therapy. The treatment is different in these two groups. I want to stop for a minute and make sure that you're comfortable with what I'm saying now. Everybody looks sort of comfortable, or asleep. Anyway, so subgroup analysis. So suppose you're interested, by the way, this is so suppose that I'm interested in the relationship of diabetes and cigarette smoking in people with advanced atherosclerosis. And the reason, I can do this in a subgroup analysis of the ACAS study. And ACAS was a randomized clinical trial of surgical management of carotid atherosclerosis. And we have, 21% of the people managed medically were diabetic. And in this example is in the context of one analysis in one arm of an RCT, but the same concerns exist when comparing arms. Okay, and you might say, why are you interested in this? And I'm interested in this because of this wonderful woman, Elizabeth Barrett-Conner, one of my heroines of my career, wrote this paper back in 1984, and what she found was that basically the impact of cigarette smoking is particularly harmful in diabetics. It's much more harmful to smoke if you're diabetic than if you're not diabetic. In addition, the impact of diabetes is much worse if you're a smoker than if you're not a smoker. And so basically in counseling your patients, what Elizabeth says is that if you've got a diabetic cigarette smoker sitting in front of you, you really need to whoop on this person to get them to quit smoking. I mean, this is it's important for everybody. But if you're diabetic, you really don't do it. So I wonder if this, I started this thinking about this paper from Elizabeth. And so there's this huge interaction such that estimated 65% of the cardiovascular disease deaths among diabetics could be attributed to cigarette smoking. I mean, diabetics die because they smoke cigarettes, according to Elizabeth. And so basically, there's increased risk of cigarette smoking. And so what I'm interested in doing is the time to stroke different between diabetics and non-diabetics, depending on whether they're smokers or not. And so what I did was, so if you look overall in this study and say, is diabetes bad for you? And so basically, the red line is the normals, and the blue line is the diabetics. Horizontal axis is the number of people who are bad, who have an event. And those are pretty much on top of each other. It turns out the hazard ratio says there's a 43% higher risk in diabetics, but it's not significant. So at least in this study, in the medically treated arm, having diabetes was not bad for you. And so basically, what I did was, it was a 43% increase in risk. The increase was not significant. But if I stratify by smoking status, watch what happens. Watch what happens. So here it is in non-smokers. There's only an 18% increased risk. Who cares? Who cares? If I look in smokers, my goodness, it's a 200% increased risk. Look at that blue line pull away from that red line. I mean, this really supports what Elizabeth said. I believe this. I believe this. Now, p-value of 0.02. So basically, diabetes is really bad for you if you're a smoker. So I could write this up if I wanted to. How many people think I can get this published? I think this is incredibly publishable junk. Okay? So there's a trend for diabetes to have a higher stroke rate. It was primarily among smokers, where the risk was nearly 3 times as great. This, by the way, I had a priori hypotheses. I had Elizabeth's paper. I really believe this stuff. It confirms my belief that diabetes is important, and it has important clinical implications. Because as a clinician, you need to go fix those diabetics who are smokers. You need to get them to quit smoking. You need to scare them and get them to stop smoking. Well, that's a nice story, okay? So, and so here's what really, publishable data, I think I can get this in a good journal. I know I can. I know I can. So what really happened is I lied. Okay, all of that was one big lie. Sounds good, but it's a lie. Okay? What really happened was that at first I really, I'm interested in stroke, and my old mentor used to say there are 5 risk factors for stroke: hypertension, 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 and some other stuff. Okay, and so basically I started out preparing for this lecture looking at differences by hypertension. Okay, overall it was not significant, and I looked in different subgroups. And by the way, when I was looking at subgroups, I looked at smoking. I did look at smoking, but I also looked at a few other things. Okay, I looked at bruits, alcohol intake, weight, previous enterectomy. I did subgroup analysis and all of these things, trying to find something different that I could talk to y'all about. Okay, nothing really excited me. There were 44 different analyses I did, and nothing. I didn't think y'all would be interested in any of that stuff. Kind of thing. It took me, by the way, about 15 minutes. Computers are like, computers are the curse of doing research. It's like giving shotguns to children. I mean, we need to be careful. It's so easy to do this analysis now. Okay? So what happened then is, cigarette smoking, if you look at my CP, I've done a lot of work in cigarette smoking. That's the next place I looked. And it was non-significant. I did the same 44 subgroup analyses too. It took me another 15 minutes. Okay? Then what happened was diabetes, by the way, is always number 3 on my list of important things. So then I did analysis of diabetes. This is really true. This is what I did. So then I did the analysis of diabetes. Overall, it was non-significant. I found the association in smokers. Wow, look at that. That's cool. Okay. I then remembered the paper that Elizabeth had written. At that point, I said, oh, yeah, Elizabeth Barrett kind of wrote about that back in the 80s. Okay. So then what I did was I ignored not insignificant. By the way, you didn't ask me what else was an effect modifier when I was spinning this yarn for you. You didn't say what else did you look at? And it turns out that diabetes is particularly harmful for ever drinkers. Okay, It's particularly harmful for people who had previous contralateral, not ipsilateral, but contralateral endarterectomy. Okay. It was also significant for people with bruits. It was significant in whites, but not whites. People with hypertension. This is a great one. I'd love to write this paper. It was only significant in the middle tertile of weight. So if you're fat or skinny, it's not bad for you. But the middle tertile of weight, it's bad for you. The mechanisms, can be hypothesized for only some of these strata. And overall, I did more than 100 analyses to prepare for this. Don't feel like this was hard. This was easy. I mean, that's the danger in this. Very easy to do. Okay, it took me about 40 minutes. I reported only things that confirmed my biases, my sense of the literature. Was I cheating? Okay, the answer to that is yes, George was cheating. Okay, George was cheating bad. How bad did I cheat? And so what I'd like to do is show you this graph. And this is one of these points I'd like you don't need to remember exactly, but the next time somebody does this, say, what did that picture look like that George talked about? So what's on the horizontal axis is the number of comparisons that you made. And what's on the vertical axis is the chance of finding at least one significant finding. Okay? So if I do one test at the 0.05 level, I have a 5% chance of finding something significant. But if I look at two subgroups, then basically I have a 5% chance and a 5% chance. Now that's really wrong, but only by a little bit. Really I have 0.95 times 0.95 minus 1. And so basically you think of it as 5 times. But if I do two tests, basically I'm already up to 10%. By the time I do 20 tests, I have a 60% chance of finding something. Okay? If by the time I do the 100 tests that I did in this flagrant cheating exercise for you, I knew I was going to find something I could make up something about. I mean, the chance that I found nothing of interest was not there. I mean, you can always find stuff. I mean, there's almost a perfect correlation between the street temperature in Tokyo and infant mortality in America. Okay, and the way I know that is you just took 1 million variables and you correlate 1 million variables with 1 million variables, and 5% of them are going to be significant by chance alone. By the way, that's what 0.05 means. And then I can start trying to say, why does the street temperature in Tokyo, it's inverse, by the way. When it's hot there, why does that prevent infant mortality in America?
So a very similar thing happens in genetics, but that's why they decrease the p-value to the times 10 to the negative 8, right? Because they are...
And what is the justification for that 10 to the minus 8?
I think that was the...
I can get you in deep trouble right now. But yes, that's exactly what, that's called the von Neumann adjustment.
Yes.
To do that, what you've done is assume there's no correlation between any of your chains. Let me say that again. Your assumption is no correlation between any of your chains. And then as the chip sizes are expanding, what impact does that have on the 10 to the minus 7? It was 10 to, back when chips only did, I'm going to say the wrong numbers, but back when chips only did 2,000, genes, we did 10 to the minus 7. Now we're doing 100,000. So what has our response been to the fact that we're now testing more genetic loci? Nothing. We're still using 10 to the minus 7. This is one of the most thoughtless parts. I mean, if you want to talk about something that's not really based on rational thought, I mean, basically what geneticists have done, hit my hand, I'm sorry.
It's okay.
What geneticists says, oh my God, what am I going to do, 10 to the minus 7? Okay, everything's fine. That's really, do we want to do research like that? I don't do a lot of genetic stuff. But anyway, so I knew I was going to find something I knew I could explain it to. Okay. Now, the cool thing about this is I flagrantly cheated. They remain highly publishable. And so if a subgroup is naturally important, how do you do it? And this is another one of these take-home messages I'd like you to have. Have y'all heard of Hill's criteria? By the way, it's Hill's considerations. Read his paper. He does not say criteria. He says considerations. One of them is that basically is the underlying mechanism. For something to believe something is causal, it needs to have an underlying mechanism. The mind is such a great tool. Once I see the association, I can almost always come up with a reason for why it's there. Almost always. So at the end of ACAS, we randomized people to inarterectomy versus medical management. What we found, complete surprise, completely unhypothesized, was that the surgery was much more effective in men than it is in women. Inarterectomy is a much bigger treatment effect in men. And my medical mentor, godparent of my children, literally, person who had huge influence on my career, said, by the way, from a different era, says, I know why this is happening. He says, that's because women, in his word, have more delicate arteries. He meant smaller, okay? Have more delicate arteries. And that surgery in women is harder than it is in men. And that's why we're getting more peri-procedural events in women, and that's why it's not as beneficial in women than men. And we were drinking wine and having a good time, and I said, gee, Jim, that's interesting. I said, but what would you have said if it had worked in women but not men? Then I would have said, men have more advanced atherosclerosis than women, and doing surgery in a messier artery is at higher risk, and that's why it works in women but not in men. And the point is, once you see it, I dare you, the next time you just look at any paper, next time you read a paper, and they have this whole paragraph about why the effect is there. So play the game with yourself and say, if it had come out the other way, how would I have written that paragraph? And I guarantee you, 9 out of 10 times, you can come up with references and all kinds of junk that says, I know why this happened. So don't believe it once you see it. Only believe it if you said it before you saw it, and then you went out and found it. Okay? That it makes sense doesn't matter. Doesn't matter. So the only way you could really stop this danger of subgroup analysis is limited. Don't do a lot of it. Be thoughtful about what you do. Plan it. By the way, with today's computer, it's also incredibly easy to forget the peaks that you did. You just say, do a correlation matrix, look at everything, pick out these things. If you do a correlation matrix between 10 factors, you've just done 121 statistical tests. You picked out the 3 things that you thought were important. Well, should have been 5 or 6 things that were significant. You only got 3. Do you believe those? Importantly, appropriately report results. There's nothing wrong with fishing in the data. Going in and just say, what's in here is a great thing to do in a hypothesis generating mode. As long as you wrote in the paper, I looked at 120 things, and these are the things I found. I hope somebody else has a data set and can confirm my findings, but these right now need to be interpreted with caution. Nothing wrong, that's good. It's only good if you did what I did, which is to play the game, because I guarantee you, you make up junk to support anything. And then if you say, report how many tests you actually did in your paper. And if the other people don't report what they looked at, watch out. Watch out. If you're a consumer, again, watch out. The most logical, clinically important subgroup analysis may well have only been one of 10 that was significant. And look to see if the authors noted the importance of this in a priori hypotheses. So this is an interesting one. This is Ascot. A good friend of mine did this study, and he published this. By the way, every clinical trial has this as the last figure. This is called the forest plot. And basically all you're doing is looking at subgroups. I don't know if this works. That didn't work either. So basically all you're doing is looking at subgroups, diabetics versus non-diabetics, current smokers. So when you look at that, what do you see? In particular, so what we're doing here is lipid lowering to stop people from having heart attacks. How many people think we should treat women? Women is, I can't point at it, but is halfway down for the people who aren't here is halfway down, and it's the only point that's clearly to the right of the line of no difference. Is this evidence that we should not treat women? Because the guy who did this, Bruce that did this, said when he got this, he got an email from this guy that says, "Oh, I see your study that you should not treat women or diabetics with this drug." Probably wasn't enough, exactly. And in addition, if I look at this number of subgroups, by chance alone, one of them is going to go in the wrong way. That's what chance is. Okay? And so, is it less effective in diabetics? And this, what I did was, I looked at 18 different strata. By the way, what's not on here is the p-value for interaction, but none of them were significant. And does this help you understand the findings? And it does for me, because what it shows for me is not that women are that different, but generally, this thing works in all the different subgroups and works about the same way. So, efficacy versus safety. Are y'all worn out? This is okay. I'll stop talking after you a bit. Anyway, so efficacy versus safety. So boy, I wish I had more time to talk about this. This one's hard. Anyway, trials are designed to assess differences in the primary outcome. Consider a treatment for stroke, and we're looking at improves the outcome, modified Rankin score of 80% of the treated patients at day 90, the primary outcome. That's what's efficacious. Unfortunately, if this study did that, and 20% of the people had a cerebral hemorrhage and died on day 1, okay, are you going to prescribe that drug? Okay, it's efficacious. My primary outcome was significant. This thing improves the outcome of stroke. It's unfortunate it kills people, but it does. I met my efficacy outcome. So that's the difference between efficacy, what my study was planned for, and safety, which is a side effect of trying to get the efficacy. Okay? Would you recommend this treatment? Generally unsafe treatments are not employed, even when they're quite efficacious. And like I said, Vioxx, I used to take Vioxx, and it really works. It really stops your pain. It's a fantastic drug, very efficacious. Okay. Anyway, clinical trials are focusing on establishing efficacy. Phase 3, I should say. Phase 3 trials are focused on efficacy. Safety is also important, so why not have it as one of the goals? And the issue is that you're pretty clear about what you want the drug to do, but you're not clear about what the bad stuff happens is before you get started. The people who did Vioxx probably didn't sit there and say, I'm worried that this thing could cause heart attacks, before they did the efficacy trial. That was really found out as a little bit of a smell of it in the efficacy trials, but it took a long time for that to raise. And so you can't say I'm worried about anything. And different safety outcomes have different value. In other words, if I have something that has a safety outcome and it causes angina in 3% of the people, that would worry me. But if it kills 3% of the people, I would worry a lot more. And how do you weight these things? Is there a scale that says, one death is 8.4 anginas or something? And, so it really gets to be hard to weight these things. And because of that, safety outcomes and stopping a study for safety is usually a gut check. We're not going to make it to the DSMB, which is the last topic on this talk, but there's a committee in the clouds that watches you do these studies. It has nothing to do with the study. We make sure that you're not doing bad things to the wonderful, kind, and generous people who are in your study. They're the heroes and heroines of all of these studies. And, for me to stop a study, I'm in conflict because, if I stop a study, I stop my funding. I want to be funded. And so you don't want me to be the person stopping the study for safety. You want somebody to stop the study for safety who doesn't lose anything if it gets stopped. And so that's the DSMB. But they end up having to say, basically, can I sleep at night given what's happening to these people? So safety is more of an opinion. Efficacy, I've written down the formulas, know what I'm going to do. Safety is more of a gut check. So basically now we've hit the end point. So it's a hard thing. It's a hard thing. Anyway, masking or blinding, So basically the whole idea is the basic concept of blinding, masking, is that I know what the study should do. I wouldn't be doing it if I didn't have a pretty strong opinion about this drug being valid. And so basically, so you're in my clinical trial, and I think you, that you've had a heart attack, or you've had chest pain. Was it seen by? And I know what treatment you're on. Did you go see a doctor? But you're on the placebo. We need to make sure that you go see the doctor. Because after all, if you were on active treatment, I know you're treated. And so basically, those biases that the investigators have, and boy, I have them, it can really bias the outcomes. And people, both the patients and the investigators, assume that the active treatment is better than the placebo. Trust me on this. One of my favorite cartoons of all time. Member of the placebo group.
That's terrible.
But anyway, physicians really want to help people. I mean, good. I'm glad they do too, by the way. And so the evaluator knows the assignments. They're going to look more intensely at the group assumed to have worse outcomes. Are you sure you haven't had a problem? And so the only way to stop that, by the way, the other thing that happens is there's differential assessment. You can find more events. For example, if I'm doing a caloric restriction study and I have weekly classes for the people who are getting caloric restriction and the other group doesn't have a weekly class, then I'm going to see the people on caloric restriction more often. And every time I see it, I can say, oh, you don't look good. We need to have you checked. And so what you have to do in these studies is be extraordinarily careful to treat both groups exactly the same. Surgical follow-up is a great example for us. If you get randomized to surgery, the stupid surgeon wants you to come back so they can check you. Well, that means I got to get the placebo person to come back and be seen by a neurologist at the same time. Because the surgeon might notice some, face drooping and then go put the person in a CT scanner and find out they've had a stroke. But if the person in the medical group is not being evaluated, then his face is drooping too, but he doesn't get put in a CT scanner. So basically, each visit is an opportunity to discover events, and the patient can't have a fever if the temperature is not taken. Okay, so you need to take the temperature the same way in both groups. So both groups, you do that by having the physician, really everybody, not know what treatment you're on. It implies that the follow-up schedule is identical. The old term is blinding. People in the Eye Institute have objected to the idea of blinding people. I'm serious. I'm serious. They don't like the idea of saying I'm doing a blinded study. They talk about masking. And so if you want to be sensitive or you want to get funded by the Eye Institute, you're doing masking. Okay? Double mask means neither the patient or the investigator knows the treatment. Single mask doesn't really always mean the same thing, but usually it means the patient doesn't know what they're getting, usually. Challenges to masking, it's not always possible. Residual surgical wounds. I mean, if you're doing inarterectomy versus medical management, even I can tell who's had an inarterectomy. I mean, even I can. Responses to treatment masking. If you're doing a study and it's a lipid-lowering drug with PCSK9 inhibitors, and there are local labs done on lipid values that are seen by the clinic staff, you might as well just stamp on their forehead active treatment and not active treatment. I mean, because that drug knocks lipids down so far that anybody who sees a before and after lipid value will know who's on what treatment. And so one of the interesting challenges in doing these studies is if it's something that is measurable, that unblinds the study, how do you take that out of the clinical management of the patient? So right now we're doing a study where we're randomizing people to stenting versus medical management, and we want to get a baseline MRI scan. Because what we want to do is also measure silent endpoints or subclinical endpoints. Well, what are we going to do with that MRI scan? Are we going to let the local clinic see that baseline MRI scan? They are going to treat the patient differently if they see that scan and there are holes in the guy's head versus they don't see the scan. Okay? And so what we have is so complicated. We spend hours working on this stuff. So in every site we have a blinded and unblinded investigator. And the unblinded investigator gets to see the MR scan because after all, they could see a great big, cancer in the guy's head. And we don't want that to go running around with no intervention. So it has to be reviewed, but we sure don't want it reviewed by the study staff. So we've got to get it out of the hands of the study staff. And it turns out these studies have 120 sites, and figuring out how to do that is no small task. You'd spend a long time trying to get the responses to masking. Another good example is when I was part of the PEPI trial, postmenopausal estrogen progestin trial, way back when, the cycling for people on HRTs who get randomized to estrogens, unopposed estrogens. Some women will notice when they get assigned to unopposed estrogens. And so they know what treatment they're on just because of the outcomes of the treatment. Serum or other assays. If it's an exercise study, how do you blind? And so part of this is to at least mask the rater. Okay, this is called a probe design, prospective randomized observation blinded endpoint. Okay, so I can't mask who got surgery, but I can have the person who's deciding whether or not a stroke happened not know what group they're in. How do you do that? Well, what we do is we go get the medical records and bring them back, mark out everything in the medical records that could possibly be related to the therapy, and then have it reviewed by a panel who doesn't know what group they're in. So at least the people deciding whether or not there's an endpoint are blind. There it is. Prospective randomized observer blinded evaluation. You can also, by the way, move towards more, if you're worried about blinding, move towards more objective outcomes. Okay. At the extreme, you need to blind a study where the outcome is death. The 4S trial, the outcome is death. Okay. It's hard to be biased in your evaluation of is this person dead or not. Okay, that one's hard to mess up. So the softer your endpoint, when you're thinking about this, the softer your endpoint. So let's say that my endpoint is a modified ranking scale, physical functioning. Oh man, whether I can move my arm or not. Now I need to worry more about blinding. The softer the endpoint, the more you have to worry about it. So finally, the fun things for statisticians, analytic issues. Are y'all comfortable? By the way, how many of y'all have heard intention to treat us? Does everybody know what we're talking about? Do I need to do this? Yeah, I am going to do this. There's one. What are you talking about? There's one. Anyway, so assume I have 100 patients randomized to 2 groups. I have in those 10 patients announced that the only reason they were in the trial was to get a stent, and they now demand the stent. They were randomized to the other group. By the way, this happened to me. The only way you could get a stent was to be in the study. Okay, way back when. And so do we compare the people getting the stent, the 110, to the people not getting the stent, the 90? Or do we compare the 100 to the 100? And I wouldn't be talking about this if it wasn't comparing the 100 to the 100, which seems nuts at the first point. And if you really, and I've picked on surgeons so much, I'm so sorry. I work with surgeons, but if you want to really upset a surgeon, it's when somebody is randomized to their therapy and they don't get it and they still have an event. And it counts against the surgery. It drives them nuts. You ought to see them. They don't deal with this well. They don't deal with this well. But anyway, but we do analyze the 100 versus the 100, the idea of intention to treat. On the surface, it makes no sense at all. There are only patients in the treatment who get the treatment can make a difference. However, what you're really doing, let me just forget the slides and talk about this. What you're really doing is protecting randomization. After I randomized the two groups, the only reason you were in this group instead of that group was that my coin ended up being heads. Now, 10 of these people move over to this group. Now other things can happen. Let's say that those 10 people who wanted to change groups because they got stents were all young, well-educated people who had read the literature on stents, and they really wanted to get it. Well, that is not a random subset of this population. I've got both education and age acting with me now. So now I've made this group both smarter and younger. So now these two groups are not the same. Now, and so what you're trying to do, all you're trying to do is protect randomization. I'm going to skip over this, but you might say, well, gee, that's unfair. If you didn't get my surgery, why should you count those events? You can do a per protocol analysis also. Usually it's a secondary analysis. And there what you do is say, if you did what you were supposed to do in the protocol, we're going to count you, and if you didn't, we won't. It's usually done as a secondary analysis. You can remember this, and this is not in the slides, but if you're doing a non-inferiority trial, a reasonable proportion of investigators think the primary analysis should be per protocol. The primary analysis should be per protocol in a non-inferiority trial. I disagree with that. Probably a third of the people think that it ought to be done. I mean, this is something you get in fights about. This is one that people, no matter what you say, you're doing it. But anyway, so that's the whole idea of intention to treat. Now I can fast forward through this a little bit. I want to spend a little bit of time talking about statistical power, changing power. So when you design this study, what you want to do is if the drug really works, it's important to say it really works. Okay? And so when you're doing this, I have the truth. And I keep pointing at this, and I realize not only for the people who are not here, the pointer that allows me to point at this is also not working. I apologize for the people who are not here. But in truth, there either is a difference or there's not a difference. We spend 95% of our time as scientists worried about the column that says there's no difference. We live in this column that says there's no difference. So we start out with the null hypothesis, there's no difference. Okay. So I do a study and I do a statistical test, and I either conclude there's no evidence of a difference, I fail to reject my null, or I conclude there is a difference. Now, if I conclude that in truth that there's no difference, and I say there's no evidence of a difference, I got it right. I win. Yay, got it right. Okay. On the other hand, if I say that there is no difference, and I say that there is, I lose. I got it wrong. Okay. We call that a type 1 error. That is the alpha, the type 1 error. What is the chance that if nothing is going on, I say that something is going on? Okay, normally 0.05 is the alpha you live with. Okay, now, what the idea of statistical power is the other column. What happens if the alternative hypothesis is true? Now, if the alternative hypothesis is true, and I say that there's no evidence of a difference, I lose. The drug really works, but I say it doesn't. Okay? I fail to reject. Alternative, and that's called, by the way, the guy who made this up spent all night trying to figure out what to call this, a type 2 error. Okay? So basically, a type 2 error is when I mess up by saying nothing is going on when something really is. However, if there is a difference, and I say there's a difference, I win. And that's the power, okay, which is 1 minus the type 2 error. 1 minus the type 2 error. Now, it's fascinating to me, and I forgot whether this is on the slide that's coming up. If it is, I'll just fast forward through the slide. 0.05 has been handed down from on high as the value that you want to use for the type 1 error. For reasons that I don't really understand, either 80% power or 90% power has been handed down by the same guy or gal for saying this is how much statistical power you have. So when you say that your study has 80% power, you're saying that if a difference is really there, I have an 80% chance of seeing it. If your study has 90% power, you're saying if the difference really exists, I have a 90% chance of seeing it. One of the things that you think of your career, by the way, for the rest of your research career, as being a series of 5-year periods. Okay? And you only have so many, I only have like 3 more 5-year periods. I'm old. Okay? And think, before you do anything, stop and think, is this the way I want to spend one of my 5-year periods? Because you don't get to spend it but one time. And if you want to do a study that has 80% power, which many people do, I think you should be required to say out loud, if this drug doesn't work, I have a 1 in 5 chance of not seeing it 5 years from now. Okay? And you ought to be comfortable with that. You ought to be comfortable with that. I'm not saying it's a bad thing to do, but I sort of question even if 90% power is too much. But we'll come to why you do study with 80% power in a minute. Okay? So type 1 error, saying that the hypothesis is false, but in fact it's true. You need to decide how much risk you want to take, normally 0.05. Okay, type 2 error, failing to see that the null hypothesis is false when it really is false. How much risk do you want to take? Again, normally 0.1 or 0.2, which is a 10 or 20% chance, which is a 80 or 80% power. So if the hypothesis is false, you need to say it's false. Beta is the chance of making this mistake, so 1 minus beta must be the chance of getting the right decision, and that's the statistical power. I want you to go home remembering what statistical power is. This is one of the points you ought to take home with you. Okay? So now, if you think about designing these studies, designing a trial, there are 5 interrelated values. If you give me any 4 of these, I can tell you what the 5th one will be. So the first one is the level of evidence required to declare significance. Okay, that's I'm going to call alpha, which is normally 0.05. You don't get to pick that very often. The next is the likelihood of seeing a significant result if one really exists. That's called the statistical power. It's 1 minus beta, and that's the statistical power. The third is the true difference between the treatment effects. I want to stop for just a minute and say, normally in a superiority trial, there's one way for the null hypothesis to be true. There's no difference between these 2 treatments. That means A is equal to B. There's only one way for A to be equal to B, and that's for both of them to be the same. There are an infinite number of ways for the alternative hypothesis to be true. If I say these 2 things are different, they can be different by a little bit, or they can be different by more, or they can be different by a lot. Okay? So the true difference between the treatments is something that can vary. By the way, in life, as in statistical testing, big things are easier to see. Elephants are easier to find than mice. Okay, I'm going to call that delta, which, by the way, is first kin to the delta that we had back in the other line. There's the sample size. You might say, why would I ever do a study with 100 people in it instead of 200 people? 200 people in it instead of 100 people in it. Okay, sample size is the currency of power. It's how you buy power. You buy people, you purchase people, and what you're getting is statistical power. It's the currency of power. And I stayed up all night, I'm going to call that N. Okay? And then finally is noise. What is the noise in the system? Why do you care how you measure these things? Why do we spend some... Well, by the way, people doing cardiovascular trials, you end up taking the blood pressure 3 times and then averaging them. Why do you think you're doing that? Just taking blood pressure is fine, right? Now, the reason for that is if I take three things and I average them, I'm reducing noise. I'm getting a better estimate of what the blood pressure is. Anything that I do to reduce noise helps the system. Basically, if I have a night, that's why, by the way, animal models are cool. You remove noise. You don't have to mess with these people things, okay? And the noise I'm going to call sigma, okay? Now, if you tell me any one of 4 out of the 5 of those, I can calculate the 5th one. And so basically, if I know, if I don't know the power, but I know the other 4, then what I'm doing is I'm going to perform a power calculation. If I don't know the delta, but I know the other 4, then the calculation I'm going to do is called a calculation of what difference can I detect? What is my detectable difference? If I don't know n, but I know the others, then what I'm doing is a sample size calculation. But it's the same formula. These are all just, five variables with one unknown. Okay, and I'm not going to go into how to do it. I wish we have 2 lectures on this in the course, but anyway, but just for a minute, if I require more evidence to declare a significant difference, what do you think happens to sample size?
It's going to increase.
It's going to go up. If I require more power, what happens to sample size? It's going to go up. If there's a bigger difference between the 2 treatment groups, what happens to sample size? It goes down. If there's more noise in the system, what happens? Sample size goes up. But it's the same formula. If you want to know the formula, I'll write that for you after the class. It's really easy. But we don't tell people because this is how we get hired. We keep this as like a secret family formula. Yes, sir.
So the noise is represented by the standard deviation.
Right.
The variation of.
Right. And so let's say that what you're doing is a trial for lowering lipid levels. If I have the same person I measure lipids on today and tomorrow, I'm going to get different values. And so what I'm going to do to remove that noise is measure it multiple times. I might measure it at 3 months, 6 months, 12 months, 18 months. I might estimate the slope instead of just taking a single value. Anything to remove noise helps. Okay. And I'm going to do one more slide because I like this one, then I'm going to quit. I'm sorry. No, I'm not. I quit.
Can we have the formula?
I'll be glad to. But anyway, I've enjoyed being here. I'm sorry I didn't get to the end of the slides. It's your fault. You asked questions. I appreciate that. The interim analysis, by the way, very quickly, this is important. Interim analysis, let me just do the 30,000 foot interim analysis. Do you think it's the right thing to do a trial that's going to take 10 years to do, where, in fact, you knew the answer at 5 years, but you didn't stop the study? Okay? So you kept on going, you put people in the study, you exposed them to the bad treatment. So that's bad. You don't want to do that. So what we have to do is to figure out a way that we can look at the data halfway through and stop the study. I already know it's either working, efficacy, or I'll never get futility. Efficacy, futility. Okay, every time I look at an answer, I need to pay a penalty. The more times I look, the more likely I am to find something. We talked about that in subgroup analysis. Same idea, same idea. Okay, so what I need to do is to figure out what penalty do I take for looking at the data. Now, I love the names of certain things. The name that this is called in our literature is called the alpha spending function. Isn't that a great name? So basically, I've got 0.05 in my hand, and I want to look at the data. Well, how can I do that? A little bit of my 0.05 and spend it. Okay? And I want to look, by the way, I want to look 3 times. The more I look, the more of my 0.05 I'm spending. Why do I care about that? Well, at the end of the study, I don't have 0.05 in my hand anymore. I might have 0.04 left, which means for me to declare something significant at the 0.05 level, it has to be less than 0.04, because I've spent 0.01 of my 0.05. Okay? And which means, by the way, if you get a study and the p-value is 0.045, you're going to write a paper that says there's no difference between these two groups, p equals 0.045. Then you're going to spend the rest of your career trying to explain to clinicians who haven't taken the time for courses like this why 0.045 is bigger than 0.05. Okay? And so you need to think carefully and plan how you take these notes. People have spent a lot of time trying to figure out the best way to spin in their alpha. And probably the best way is something called O'Brien-Fleming. And then O'Brien-Fleming is the most common. But it all comes in. And then the very last thing to make, which is in the slides that y'all have, and I hope you do, is the idea of adaptive trials versus interim stopping. Just very, very quickly, interim stopping is I plan a big study and I try to stop it early. Okay? An adaptive trial is ill-defined. Most people, when they say that means I plan a small study and I decide if I have to make it bigger. Okay? Hot topic. Okay, adaptive trials make you sound smart. In my humble opinion, you're not smart if you do an adaptive trial. Okay, all it does is make you sound smart, make you feel good, but it's not a good idea for a lot of reasons, and I wish I had more time to talk about it. So be careful on this idea of adaptive trials. If you want to do one, give me a call. So I'll talk here for free. It's going to cost you a beer to get that part of it. Yes, ma'am. By the way, the lecture's over. Leave if you need to leave. I'll stay here for a minute if there's questions. I'll wear this mic just in case somebody wants to type in a question.
I'm interested in formula.
The formula. Well, I don't have this on my computer. Okay. This might not be Z plus Z of 1 plus beta is equals to delta divided by sigma divided by the square root of n. Now,
Just plug it in.
just plug it in.
Supposedly.
No. So this, by the way, is always 1.96. This is either 1.65 or 2.1. This is 90% power. So this is the true difference. This is how much